Righty. Hello, hello. How is everyone doing today? Hopefully well. I am at a undisclosed off-campus location. No, I'm at the Oakland West Center, which is kind of, hey, it's west of campus, right? You go down University West, you go over 75, you cross Opdyke, and then it's on the left. So that's where I am. Um, there's heat. That's cool. Um, Yeah. So, and I'm obviously this is I'm meeting students that need to make up exams and whatnot. So that's what this is for. So, speaking of exams, I have news. I literally just got it. So you know how I think on Wednesday, um, is that when we met? Yeah, on Wednesday, um, I had said at that point that the plan was for Oakland to confirm with faculty by the end of business yesterday, right? 5 p.m. Thursday. Oakland was supposed to have sent out an email letting everybody know what the accommodations were going to be for final exams. Well, of course, 5 p.m. came and went, and then 6 p.m. came, and then 7 p.m. And um, eventually, I forget what time it was. They did send an email, and it was like, "You will find out early tomorrow morning." And so then, I was here at 8, nothing, no emails, nothing. I finally got the email, right? So, long story short, we are going to be, all right. Now, this is I got to make sure we are the 120 class, right? And we are CRN 43721, right? Okay. So our final exam is on Wednesday, right? December 10th. Let me just make sure that my dates are correct because I don't want to misspeak because that would suck. Yes. Okay. Wednesday, December 10th. All right. Um, at 3:30, which is when we were scheduled. Um, we are going to be in the Human Health Building, room 4050, HHB 4050, 4050. So the Human Health Building is on campus. It's a newer building. It's kind of at the corner of it's along Squirrel and Walton. So the northwest corner of campus. That's where we will be on Wednesday the 10th from 3:30 to 6:30 in room 4050. I will send this information out in an announcement so you'll have it. But we've got a heated space for the final in person on Wednesday, right, from 3:30 to 6:30. So that's good. So we can look, you could plan, I guess, on that and plan for that in your mind as you're trying to figure out how to navigate these next few days, right? Um, someone did have Emily, did you have your hand up, have a question, and did I answer it maybe, possibly?
Oh, I did, but I was going to ask where we were going to, if we had a location, and now, well, now we do.
Yes, we do. So that's all excellent. Yes. So I again I'll send out an email with this information, and I'll even include a little picture of the building so that you can kind of you get a sense of where it is if you're unfamiliar with it, right? Um, so that's good. Um, I think that was the main like logistical thing that I could think of, right? So yeah, I think that was the critical thing, right? So in terms of material, right? Remember 19, we're not going to be covering 19 at all, right? Um, and we are going to cover some 18 for sure, right? I'm gonna for 18, we're gonna cover basically just some big picture ideas about epigenetics, which is changes in a trait not due to gene or DNA mutations, right? Um, we're talking about things outside of changing the actual DNA sequence that changes gene expression that then can change traits, right? So that's epigenetics. And we'll look at one example for sure, X-inactivation. Emily, you've got another question.
Oh, also, I was going to ask on Monday and Wednesday, or Monday of next week,
Yep.
not Wednesday, because that's when the final exam is. On Monday, are we going to have a lecture on Monday?
Nope.
Okay.
And in fact, I have a morning exam that morning, so I will be busy with that.
Okay.
And then I have an exam on Tuesday morning as well. So yeah,
Oh,
um, I think moving forward, and this is for everybody. Next week, since our exam is on Wednesday, um, we've got Monday and Tuesday, right? I will be busy in the mornings those 2 days, but I should be around and available in the afternoons on Monday and Tuesday if you want to try and set up like a individual Zoom or something. Let me know and we can kind of try to work out a time where we can do that. Um, I told my 920 class over the weekend and all the way up until Wednesday. If you've got questions about material or something, email me over the weekend or whatever, and I will be monitoring my email and I will try to get back to you as timely as I can with usable information and whatnot. So um, certainly, as we move through, the rest of the time up until Wednesday, you can certainly reach out to me and we can meet and whatnot. Probably virtually. I think campus is still going to be closed until the 11th, maybe Thursday, something like that. So we will see. But again, I can be available in the afternoons on Monday and Tuesday next week, um, and email communication all along, right? So, okay. I need to get my chat. I want to show the chat. I want to be able to see the chat. Okay. All right. So, we're done with seven. I think we finished seven. Did we finish 17 on Wednesday? Yeah, I think we did. I think we pretty much did. Um, okay, good. Thank you, Casey. All right. So then let me share my screen. Okay. Participants can now see your screen. I actually cleaned up my desktop just for you guys. I'm kidding. Um, okay. All right. Can you see that chapter 18 PowerPoint slide? Hopefully, yeah. Okay. All right. Um, okay, so 18, this is all like non-Mendelian, right? Genetics is either Mendelian, and it plays by the three rules, dominant recessive, mutant, and wild type alleles, and then independent segregation, or a trait or a condition is passed on in a way that is not Mendelian. So that's non-Mendelian, right? And 18 kind of works with some of these. So let me see. Um, all right. Got a couple of chat questions. So we don't have to. So, okay. Um, extra credit or makes up for one if you missed. Correct. So, um, if you look back through the syllabus real quick, you don't have to do it now. Um, there are 17 PKA assignments, 17 chapters, 17 PKAs. There are 17 PLQs, one for each chapter, right? Um, I will take 15 PKAs, right? Those are each worth 2 points. So your first 15 PKAs are pass fail. You complete them, you get your 2 points. So the first 15, that's 30 points. That's 100% of the PKA points. That leaves two extra PKA quizzes, right? And if you were to go sequentially, it would be chapters 18 and 19, right? Those are the last two. Um, Same with the PLQs, right? If you went 1 through 19, 17 is the 15th PLQ, and then we've got 18 and 19. So there are two extra PLQs and two extra PKAs. If we're not going to do 19, and I think 19 has been open since September, and I think I even have it labeled as like extra credit, right? So if you do that, I would highly recommend you do the PKA for chapter 19 because it's pass fail, and that's 2 easy points, right? If you missed a PKA earlier in the semester, or you can't, if you're not going to get to 15, if you do that extra 1 or 2 PKAs, that'll fill in and make up for any missed PKAs up to 15, right? If you are at 15 and you do both PKAs, those are going to be 4 points that are added to your exam total points, right? So, like if you had a 42 on one of the exams and you do those two PKAs, that 42 is kind of like a 46, right? Same with the PLQs, right? If you go sequentially number 1 to 17, those are the first 15. That's what you got to do. Now the PLQs, remember, are not guaranteed 2 points, right? But they can get up to 2 points. So for the PLQs 1 through 17, right, you can get up to 30 points total, right? And then that leaves 18 and 19. Well, we've talked about 19. That's kind of the extra credit one. And then that leaves 18. Similarly, if you do your 15, and then you've got the two extra, and you do those, and let's say you get 1.5 on each of those. So that would be 3 points then that would be added to your exam totals, right? So that 42 that became a 46 from the PKAs would now become a 49 from those two extra PLQs, right? So if you missed PKAs, you can do the extras and fill in. If you missed PLQs, you could do the two extra to fill in, or do everything, get everything, and you get maybe up to 8 extra credit points, right? So that's what those are there for, right? You don't need to necessarily study the 18 or I mean 19 material for the exam, no. But you can get some extra credit that way, right? So hopefully that kind of answers questions about that, right? And again, if you want to get more information about how the extra credit for those quizzes works, consult the syllabus because I have it in there pretty clearly, right? So, okay. All right. Any other pressing matters before we kind of dive back in? Okay, cool. All right, so 18, non-Mendelian, all right? And we will look at a couple of things. We're going to look at epigenetics. Maybe we'll touch on linkage ever so briefly, just to kind of let you guys get an idea of what linkage actually is and how it's useful and how we like literally figure out where a gene is in relation to another gene on chromosomes, right? We can map them, right? And then we've got extra nuclear inheritance, right? What does extra nuclear mean? What does that word mean? Extra nuclear. Anyone want to take a guess? Is it nuclear? No. It's not in the nucleus. Exactly. So, what non-nuclear inheritance is that? Are we talking about here with this extra nuclear inheritance? If it's not the 46 chromosomes in the nucleus of the cell, it's the what? It's the exactly, Malik. It's the one chromosome in our mitochondria, right? Remember, mitochondria were their own little creatures, we think billions of years ago, and then they kind of got into one of our cells and then struck up that arrangement to make ATP, right? So mitochondria don't follow meiosis. Mitochondria aren't involved with sexual reproduction or anything like that. And so mitochondria are just going to kind of pass on their genes very straightforward wise, right? And the thing that's cool about mitochondrial genetics and mitochondrial DNA and mitochondrial inheritance is that, remember, the size of the female gamete is massive, right? The female egg is huge. And then the male gamete, the sperm, is super, super tiny, right? The sperm has got 23 chromosomes, and that's it, pretty much. The egg has got 23 chromosomes and then all of the mitochondria. So once the sperm fertilizes the egg, every cell in that embryo, as that fertilized egg divides and divides and divides and divides, the mitochondria are dividing and dividing and dividing. And so mitochondria are inherited through the mom, right? So maternal lineage, because all of our mitochondria that we have originated from the egg, because the sperm doesn't have any, because it's just the missile to carry 23 chromosomes to the egg, right? Um, and you can get mutations in mitochondrial genes, and you can have a range of symptoms, and they're usually really mitochondrial conditions are very difficult to diagnose because who's going to think mitochondrial DNA when the kid's got a learning problem or something, right? We don't think that way. So, yeah, something to think about. We're not going to look at that. I want to focus on epigenetics, right? Okay, so oops, wait a minute. So epigenetics, I moved, or I kind of um, switched the slides around a little bit. I wanted to move epigenetic like definition up because it was buried deeper, right? But when we're talking epigenetics, I need to. Here we go. I'm gonna get my pen. There we go. Whew. Okay. Um, so all right, epi, right? What does epi mean? Or what's like an example of that, right? Epi. If you think, our skin, right? Our skin, the portion of our skin that we can see is the epidermis. Just below the epidermis, the next layer is the dermis, right? So the epidermis lies on top of and is outside of the dermis. So by that logic, epigenetic inheritance would be what? This would be some kind of inheritance pattern that's due to something outside of genetics, right? So we're talking a change in the genetic material that is not a mutation, right? We're not mutating the DNA. This is critical, right? We have mutations to mutate DNA, right? And that can change gene expression. Epigenetics are processes, mechanisms, phenomenon that we have discovered that specifically change or alter gene expression in an individual, but it's not changing the genotype. We're not changing the DNA at all. We're not making any mutations. We're simply changing how a gene is expressed. Right? This is what epigenetics is looking at. Right? And the other interesting thing about, or important thing about epigenetics is that this thing, this change, this phenomenon that is going to modify the DNA and alter expression, this change is fixed in the cell and in the individual for pretty much that cell's entire lifespan as it divides and produces daughter cells, and or for the entire life of the individual, right? They will have this permanent change in their expression of genes, right? And so we get a more or less a permanent change in the phenotype of the individual. We're not changing the genotype at all, right? So looking at a genotype may not show us anything. It's kind of like, if an individual is demonstrating a dominant trait, like if a pea plant is tall, well, what's the genotype? We don't know. It's either big T, or big T, little t, right? We don't know. With epigenetics, it could be big T, but that plant is not going to be tall. It's going to be dwarf, right? Even though genetically it's big T, right? So this is kind of what epigenetics is kind of doing, right? It's going to change the phenotype in the individual, but not the DNA, not the genes, right? So that's different, right? So two examples that 18 talk about is X-inactivation and then genomic imprinting. We're going to focus on X-inactivation primarily. It's a little bit more relatable, and I've got a good example that's personal for me. So we'll talk about X-inactivation for sure. So on the final, X-inactivation from 18 will definitely be there, right? So make sure that you review, the requisite material, right? All right. So I started off by saying that epigenetics, extranuclear genetics, linkage, these are non-Mendelian, right? That's the three general rules for Mendelian genetics, right? The particle, the gene, right? The thing that gets passed on from parents to offspring, the genes, right? Basically don't change, right? Genes are passed unaltered from cell to cell, generation to generation, right? Now, do mutations occur? Of course, right? But they are very rare. So generally speaking, right, our 23,000 genes are maintained pretty well, right, in terms of their sequence. We generally don't have a lot of mutations occurring in us in our lifetime. That's really nice, right? Now, obviously, cancer is an indication of genetic change and whatnot. So it happens, right? But in a otherwise healthy individual in a otherwise healthy environment, our genes don't change. Right? We've got DNA repair systems. We lock them in the nucleus, right? So genes are usually passed unaltered. Chromosomes, genes, and then chromosomes by extension, right? Observe the law of segregation, which is during meiosis, right? Meiosis one, right? The chromosome, the homologous pair, like chromosome pair number one, they replicate, we pair up, we can cross over, right, during prophase one. And then at anaphase one, we separate the homologous chromosome pair, right? But it's independent. So if chromosome pair number one lines up and separates like this, chromosome pair number two could line up the same way, blue on my left, red on my right, and then separate that way. But chromosome pair number three could line up like this, with the pink on the left and the blue on the right, and then we separate. Right? Chromosome pair number four could line up like this, but then chromosome pair number five lines up like that, and chromosome pair number six lines up like that, and then seven and eight, and right. That's the 2 to the 23rd power. That's the 8 million combinations, right? And this is during meiosis, right? And then we saw when Mendel did his dihybrid cross, right? When we looked at seed color, yellow or green, and seed shape, smooth or wrinkled. Each heterozygote then can make four possible gametes, four sperm and four egg. And so then you have those 16 possible offspring. And Mendelian genetics tells us that the ratio of those 16 offspring is always 9 individuals will be dominant for both traits. 3 of the 16 will have one dominant trait and the other recessive. 3 of the individuals will have the recessive trait and then the dominant trait. And then 1 individual will have both recessive traits. All right. That is borne out by independent assortment. And if you go back, I don't know if maybe we did this. I hope we did this. But if you were to count the number of individuals in those 16 possible offspring, right? If you counted of those 16 offspring, how many were dominant color? How many of those 16 were yellow? You would see that it was 12, right? 12 out of 16. Well, if we reduce that fraction, what is it? 3/4. All right. And if you look at those 16 offspring and you look at seed shape, smooth versus wrinkled, what do you think we're going to see? You're going to see another 12 individuals out of those 16 that are dominant for shape, smooth, right? And that also is 3/4, right? 12 out of 16. So that right there confirms, right, that seed color will segregate into the 3-to-1 ratio, 3 dominant smooth to 1 recessive wrinkly. And well, if that was color, 3 yellow, 1 green, and then seed shape, 3 smooth to that 1 wrinkled. All right. So within the 16 offspring, the 3-to-1 ratio is still there for both traits. That confirmed that more than likely seed color was on 1 chromosome pair, seed shape was on another separate chromosome pair because they were fully independent. Okay. Again, with Mendel, we can predict. That's what's important, right? If we know genotype, you can almost always predict phenotype. Tall, tall, dwarf. We know this, right? That's Mendel. That's good, right? What are these things? What epigenetics, right? Non-Mendelian inheritance, it breaks the rules, right? What are the rules? So, well, we just talked about the rules. How do they break it? So epigenetics breaks rule number 1, right? Rule number 1 being genes don't mutate, right? They don't change over time much. And so therefore the expression of a gene should not change much over time because that gene is not going to get mutated, right? Because we protect our DNA and we fix our DNA. Well, again, epigenetics changes the expression of a gene without changing the DNA. Right? So we're not touching the DNA at all. We're just touching the expression of the gene. So that's kind of breaking rule number 1. You can have a dominant allele, big T, but it's not expressed for some other reason and not because it's a mutation, right? That organellar inheritance, right? Mitochondria. And what's the other thing? What's the other organelle that has its own inheritance? Mitochondria is one. What's the other organelle? Yep. Chloroplasts, right? They've got their own chromosome. So mitochondrial inheritance and chloroplastic inheritance are completely non-Mendelian because those guys don't have anything to do with meiosis or mitosis or anything, right? So that clearly breaks rule number 2, right? And then rule number 3, right, with seed color and seed shape, moving from parent to offspring independently right? So that we have the again we have this, right? We have the overlapping 12 and 16, right? The three dominant to the one recessive, right? For each of those traits, right? Um, if the seed color gene and the seed shape gene were like right next to each other physically, we would probably call them what's called linked. And so then they would be inherited from parent to offspring as a group. So the two alleles of those genes would always be moving into the offspring as a group, right? The closer two genes are, the smaller the chance of a crossing over event occurring in meiosis and breaking those two genes apart, right? Because if they're really close together physically, this whole chunk, right? Maybe this whole chunk of the chromosome is going to be exchanged with this one. So those two genes really close together are always going to be inherited as a group. That's called linkage. We'll maybe get to a little bit of that at the end, right? Okay. Um, oops, what are we? So again, we are changing gene expression, but we're not changing DNA, right? And so how do we do this? How do we regulate, how do we change gene expression without mutating the DNA? Well, we've already looked at some examples, right? We can chemically modify the actual DNA molecule. We're not mutating the DNA, right? If we add methyl groups, right, CH3s, right? And if you recall, if we think back, chapter 12, in our promoters, in some promoters of our genes and some gene promoters. We have what are called those CPG islands, which is literally in the promoter or near the promoter region of a gene. Is going to be a long stretch, relatively speaking, of C's and G's, those bases. That CG stretch is a target for methylation, right? And when we methylate, I think it's the C. I think the cytosine is what gets methylated, not the guanine, but the cytosine is what gets methylated. When we methylate these bases, the DNA, right? Again, if the DNA is usually like long, skinny spaghetti, right? When we methylate DNA, it starts to pack up, right? And it gets bound up. And guess what? Bound up, compacted DNA, we can't express this. All right. So this is a way to turn a gene off. We methylate the DNA, we turn the gene off. We're not mutating the gene. And if we just so happen to methylate the promoter to turn that gene off, we then maintain that methylation. So the gene is off forever in that cell or in that individual. Right. That's epigenetics. Right. It's long-term maintenance of this change. Right. So long-term maintenance. Yep. Now, everything, most things can be reversible, right? Most things, there are always exceptions, right? So nothing is forever, truly, right? But some of these gene expression epigenetic changes can be relatively permanent during the individual's life, like it's set and it will not change, right? Others can be reset, right, or are reversible. Right? Various mechanisms that scientists have appreciated that can alter gene expression without changing the DNA sequence, right? We talked about methylation. Again, chromatin remodeling, right? The beads, the nucleosomes, right, that we wrap the DNA around, we can modify, we can phosphorylate, right? We can acetylate those histone beads. And by chemically modifying the beads that we wrap the DNA around, we can once again go from a nice open form to a nice packed up silent form, right? We can't express that DNA. So modifying the histones, we pack up the DNA, right? Remember, this form of DNA, the chromosome, is very compact. This is generally speaking transcriptionally silent. All right. When chromosomes are fully packed up in their smallest shape, right before we separate them in mitosis or meiosis, these chromosomes are pretty much silent, right? Because they're all packed up. Right? But we can pack up individual regions of a chromosome really tightly. And so then that region of the chromosome and any genes in that region that's extra tightly packed up is silent. And if we maintain that packaging, those genes are always silent. That's epigenetics, right? All right. So a really a good example, a relatable example for this uh, epigenetic regulation of gene expression is X-Inactivation, right? And this is only in females, right? Why is it only in females? Well, because females obviously have two X chromosomes as your 23rd pair, right? So women, you have two homologous X chromosomes for your 23rd pair, right? This copy of the X and this copy of the X, they have the same genes, might have different alleles, right? Because you're getting one X from your mom and you're getting the other X from your dad. So whatever genes are on the X, you could have, two copies of them, women, right? Or you could be a homozygous and have the same copy on both, right? Who knows? You have to do your genotype to find out for each gene, right? So women, you are fully homozygous and you have homologous paired X chromosomes. Right? Males were one, right? We have one X and one Y. Now, are these chromosomes the same? Of course not, right? This is pink and this is blue, if nothing else. This is big, this is tiny, right? This is the X, this is the Y in a male, right? Or it could be the other way around. It doesn't really matter. It doesn't have to be the pink, right? This could be the X, this could be the Y. Um, the genes on the X are not on the Y. The genes on the Y are not on the X. So men, we have one copy of every gene on the X, and we have one copy of every gene on the Y. These two chromosomes are not the same. So these are not homologous chromosomes. We call them hemizygous. They're kind of similar, right? Males are hemizygous for our 23rd pair of chromosomes because they're not homologous, true, because it's an X and a Y. But for meiosis, for making eggs and for making sperm, the X and the Y act like homologous chromosomes, right? They will each replicate and then pair up during meiosis one and then separate, right? Just like the other 22 pairs, right? Okay. X inactivation. Well, we need the genders to be equal. Evolution wants equality, right? That's really what it wants. And so what we've discovered is that in every somatic cell, so that's the body cell, right? Not egg or sperm, not gametes, but in body somatic cells, it turns out all X chromosomes are turned off except one, right? Now, why do I say all X chromosomes are turned off? Because humans can be born with the whole range of sex chromosome abnormalities, right? Turner syndrome, Klinefelter syndrome, triple X, right? There are multiple conditions of live living humans who have a variety of combinations of their sex chromosomes, right? So that's why I say all but one will be shut off. Now, in a normal female, in all of her somatic cells, there are going to be the two X chromosomes, right? Because she's normal. And so then in every one of those cells, one of them is turned off, right? In every cell. But here's the deal. If this chromosome is on in one cell, this X chromosome could be on in another cell. And then this one is in one cell, and this one's in one cell. And so each body cell is randomly has an X chromosome inside of it, randomly inactivated. Right? Which then means in a woman, in your body cells, right? If we were to look at which X chromosome was actually being expressed in your cells, in some cells, it's going to be the paternally derived X, the one that you got from your dad. And in half of your cells, it's going to be the X that you got from your mom that's going to be being expressed. All right. That is X inactivation. And this is called the Lyon hypothesis. Dr. Lyon, I believe she discovered this concept, this process. And the hallmark of X inactivation is when you look at a cell and you look at the nucleus and you use special dyes and stains and whatnot, what you can do is you can highlight and label the active X. Right? Notice it's kind of diffuse, right? And it's kind of dim because it's all kind of stretched out spaghetti-like, right? But then look at this bright, bright orangey yellow thing that's attached to the wall of the nucleus, right? That is called the Barr body, Dr. Barr. And the Barr body is the X chromosome that has been packed up and inactivated and then just shoved to the side. And it just sits there and it stays there and it stays compacted. When this cell divides, this guy is replicated and then split. And so then the two daughter cells are inheriting the same X chromosome that was inactivated. Right? So it's maintained. That's epigenetics. We do it once and then it's maintained. Right? Okay. And it's methylation, right? That's how we pack it up so tight. We methylate the DNA. Right? Now, what's the real life example of X inactivation that you might be familiar with? If you're a cat person, maybe you've had a calico cat, or you've probably seen them. I am a cat person. Right now, I have a tuxedo cat. That's a black and white cat. Oreo, or Dog. That's her nickname. She's a great cat. And then her sister is all black, and she's Noir. Right. So those are my cats now. Before these two, I had sisters, Monkey and Minky. This is a picture of Monkey. They were both calicos. They were sisters. Minky got sick when she was 3 or 4, and we spent like $3,000. Did you know you can give cats and probably dogs as well? You can get them. They can get blood transfusions. You know that there are pet owners that take their pets to vets and animal hospitals, and they draw blood from the pets to then give to sick pets that need blood. That blew me away. And so, yeah, so Minky had a couple of blood transfusions and it didn't help. So we had to put her down. So that was really, really sad. But Monkey survived. Monkey was a tough little cat. She's calico, right? Calicoism is a direct result of X inactivation, right? At some point after fertilization, right? So in Monkey's mom, when the egg and the sperm came together to form the zygote, the fertilized egg that would become Monkey, one cell, 2, 4, 8, 16, 32, 64, 100, right? We start to double. At some point, the X inactivation process kicks in, and then in every cell that is not an egg or a sperm in the individual, and Monkey was a girl, so in all of her cells except her eggs, randomly one or the other X was turned off, right? Well, it turns out hair color is on the X chromosome. So hair color in cats is a X-linked trait, right? And here's the convention that we use, right? Rather than just using like an O or a B, right? Or a C or a T, right? Like what we have been using, right? When it's a sex linked or X linked, we use the actual chromosome and then we put the allele, right? So on the X chromosome, there's the hair color or coat color gene, and there's only two possibilities. You either have the O allele or the B allele. Right? The O allele tells that skin cell to make an orange hair. The B allele is the instructions for the skin cell to make a black hair. All right. And so now, only when you have your two X chromosomes, one containing the orange allele, the other containing the black allele, only when you have that specific heterozygote, an orange allele and a black allele in a female, right? Then randomly in every somatic cell, either the black allele is turned off because that X is inactivated, or the orange allele is turned off because that X is inactivated. And so what you then end up with is a mixture of skin cells producing orange and black hair all intermingled. And that is the calico pattern, right? Right. At some point early in embryonic development, right, the signal is somehow initiated. I don't know what initiates it. But then from that point forward, in every cell that's not an egg or a sperm type cell, one X will be randomly on, and in another cell, maybe the other X is randomly on. So then you're going to get this mixture of cells with the orange allele on and cells with the black allele on, and that produces the calico pattern. Right? Random X inactivation. Right? The white, right? The white is due to the fact that these skin cells and this part of the cat's anatomy develop earlier than the rest of these, body, skin, hair cells develop, right? So in the white, the X's have not inactivated yet or whatnot, so we don't see the pattern there. Only when we get into these cells slightly later in the cat embryonic development do we get the cells where we then start inactivating the X, and that's what then leads to the calico pattern here and not on like the ventral belly surface, right? Those cells are derived differently during development, embryonic development, right? All right, so why inactivate? Well, again, nature, we want equality, right? And I think the really important reason why is because men have one X, women have two. That's unequal, right? We can't have that, right? Because if women are expressing two copies of each gene on the X, men would clearly be at a disadvantage, evolutionarily in some way, because we could only express the one copy that we had. So dosage compensation is a really big reason, I think, why this evolved. All right, let's equalize the number of genes that'll be expressed between the two genders. All right. The other thing that's kind of cool is, and I hadn't really thought about this, is randomly turning one X on, or I'm sorry, randomly turning one whole X chromosome off, in each cell. All right, this one on, this one off, and then this one on, this one, right? Randomly activating and inactivating. The woman is going to be what's called a mosaic, right? In one cell, one half of your cells, this X is active, and maybe it's got all good genes, right? In half of your cells, this X is going to be active, and maybe it's got a couple of genes that are recessive, right? And so now in that cell, you're only going to be expressing that recessive X trait. right? Or that recessive X allele, because the other X, which maybe has the wild type allele, is silenced, right? So then in you, you're going to have half your cells with the normal protein and half your cells with the abnormal protein or whatever. And it's thought that by inactivating the one, that helps the mosaic, that helps, women, females survive with half your cells showing the dominant allele, maybe, and half your cells expressing recessive X alleles, right? So that's another reason why we inactivate, right? Now, how, right? How do we inactivate? It's I call it's a little oxymoronic, right? We want to take a chromosome and we want to transcriptionally turn it off. Right? We want to make it so that there are no genes being expressed from the X chromosome, right? That's X inactivation. So, how do we do this? Every X has got a region called the XIC, right? And that's the X inactivation center. And so the cell, and again, this is beyond me. I don't know how this works, but the cell will detect the number of XICs, and that is a representation of the number of X chromosomes. Typically speaking, the cell will detect 2 XICs, right? And what will happen then is randomly, one of the X's will be chosen to be turned off. The other one will remain on, right? If there are more than 2 X's, all the additional X's are turned off except one, right? So how does that work? Well, in the XIC, there's a gene, and that gene is called the Xist gene, Xist. Right? Now, I complain or I make fun of scientists and how not creative we are with names and stuff. And the Xist gene is a prime example of not being very creative, but it's a very interesting name if you think about it, right? So Xist stands for the X inactive specific transcript, right? X inactive specific transcript. I'm going to let that settle in for a couple of seconds, right? We want to turn the X chromosome off. We count the XICs. In the XIC is a gene, the XIST gene, and it's called the X inactive specific transcript. So what do you think that means? What's the deal with this XIST gene? Does it get turned off? Nope. This is the one gene that stays on as this X chromosome is being inactivated. So it's oxymoronic. We do need a gene to be expressed from the inactive chromosome to inactivate it, right? So how does this work? How does this XIST gene do this? Well, it's a gene, and what do genes do? They go from DNA to RNA, right? In transcription. This XIST gene literally gets expressed, and the RNA is made via transcription for the Xist gene, but then it's not translated. Right? Instead, the mRNA basically just literally coats the chromosome, that X chromosome, and that recruits additional proteins, and then the packaging and the spreading, continues, and we end up with this tiny little mass of cramped, packed, methylated, transcriptionally silent X chromosome / bar body. Right? But the Xist gene, one gene, is being maintained on the inactive X. And that Xist gene maintains the inactive state, right? And at mitosis, right, this is a cell division, right? Both chromosomes are replicated during S phase, and then we pull the bar body chromatids apart. Actually, it would be this, right? The blue and blue. We pull the bar body chromatids apart, and then each daughter cell is inheriting the exact same X chromosome that's been turned off. That's the maintenance, right? Epigenetics lasts forever, just about, right? So this is a very interesting process, right? We have to have one gene on the chromosome that we want to completely turn off. So we're not really completely turning it off, but the one gene that's on turns everything else off. But we do it randomly. Right? I'm not gonna. Oh yeah, let's do this one. This is just a quick little uh, concept check, I guess. A person with Turner's syndrome would have two bar bodies present. Is that, come on, true or is that false? What do you think? A person with Turner's syndrome would have two bar bodies. Is this true or false? The answers are rolling in, and you guys are correct. That is false, right? Remember, Turner syndrome is a human who is 45X, right? Sometimes also it might be written as 45XO or X null, right? That indicates what? One X chromosome. And so then if there's only one X chromosome to begin with, are we going to ever inactivate that? No, because that's death, right? All humans have one X chromosome, Turner syndrome, right? There's no male that just has a Y. Nope. Um, there can be males that there can be males because this is so great. This is so crazy. Because in males, the X and the Y play along at meiosis. Every now and then, the couple of genes on the Y that determine maleness, those genes can get transferred onto an X chromosome. And then you will have a human who is genotypically XX, but one of the X's is mutated, and that human will phenotypically be male. All right. If the SRY region, that's what it's called on the Y chromosome, if the SRY region specifically is cut out and transferred from the Y to the X and it's functional, that egg or sperm or whatever, that will become a male, even though they're XX. Kind of interesting. Right? So false. Right? Again, normal female, one bar body, normal male, none. Turner syndrome, no bar body. Triple X, two Klinefelters, right? That's the male with the two X chromosomes, right? You don't have to be a female to inactivate. It's just a property of biology and of life. If there's more than one XIC in the cell, we got to get rid of one. We got to silence it, right? So XXY, right? If it was XXXY, there'd be two, right? If we had a quadruple X female, I don't know if that's viable. I've only seen triple X syndrome. But if a woman or a cell had four X's, there'd be three bar bodies, right? So very interesting. Right? At least I think so. Okay. Um, that's that's the end of X inactivation material. We've still got a few minutes, so I'm gonna just kind of jump through the rest of this and don't worry about any of this material being on the final. This is just to kind of, you guys will see this. Maybe you've heard of these phrases or terms, or maybe you will see them in the future. And if you go into medicine, hopefully you'll do a genetics rotation somewhere and you might see some of these. And this is really interesting. So X inactivation is epigenetic. Genomic imprinting is another epigenetic process. And in genomic imprinting, a chromosome or a gene or something is usually chemically modified and permanently turned off. But the trick here is the region of the chromosome that gets turned off or stays on differs between the mom and the dad, or the maternal and paternal chromosomes that are in you, right? So this is again one example. So in imprinting, right? So here's chromosome, let's say chromosome one from the dad, right? These little pins represent genes that are turned off, right? We have set patterns of genes that we specifically get turned off on particular chromosomes, right? That same chromosome number one in mom, she has a different pattern of genes that just get silenced or imprinted, right? And so then in you, right, whether you're a male or a female, when you make your gametes, you impose your imprinting pattern on all of your chromosomes. So the half of the chromosomes that you got from your mom, right, you will imprint with your set pattern, right? And I don't know what creates the pattern, right? Don't ask me that. But similarly, if you're the female, right, when you are making your eggs, you wipe the chromosomes clean in terms of what genes are imprinted and turned off to begin with, and you instead impose your imprinting pattern, right, on the chromosomes. So the imprinting is still there, and usually this is methylation, right? And we're usually going to silence these regions, right? Now, it can become an issue. Mice have an issue. I guess the big deal with imprinting, again, with epigenetics in general, looking at the genotype doesn't tell us necessarily the phenotype. Or looking at the phenotype, we don't necessarily know for sure the genotype, right? Um, with mice and this IGF, insulin-like growth factor 2 receptor, right? All mammals have two copies of this gene, right? And sometimes you'll have a normal copy and a mutated or inactive copy, and they use that little negative sign to indicate that. Well, it turns out that certain chromosomes, is it the chromosome or is it the gene? No, the gene, right? Um, certain parents, right? The male parent will always express this IGF-2 allele, the wild type allele, and the female allele, the IGF-2 minus, is silenced, right? That's the imprinted gene. And so then this individual is normal sized, right? But on the other side, right, here in this example, right, the imprinting is different. In this case, the dad's normal, I'm sorry, the mom's normal IGF-2 gene, right, is silenced. And the dad's abnormal IGF-2 gene is on. So in this individual, there's still a heterozygote, but the normal allele here has been imprinted and turned off. And so now they're left with just the abnormal. And so then they become a dwarf, right? Or they're a tiny mouse. If you genotype these mice, they would be the same genotype as the full-size mice, right? That's the hallmark of epigenetics, right? The genome doesn't necessarily fit the genotype doesn't necessarily fit the phenotype. So that's mice. In humans, right, there's two conditions, Prader-Willi syndrome and Angelman syndrome. And Prader-Willi is the big hallmark for Prader-Willi is compulsive eating, like serious compulsive eating. Families that have children with Prader-Willi, they literally have to put padlocks on all of the food cabinets. They have to literally lock the refrigerators at night because the kids will just plow through a whole gallon of this and a whole half gallon of that, and they'll just, right? Because it's part of the condition. Interesting, right? So Prader-Willi, right, comes from chromosome. So we're talking chromosome 15, right? So one chromosome, chromosome 15, a couple of different regions of the chromosome that get imprinted, right? And it turns out, right, if the genes on dad's chromosome that get silenced, right, if they're the normal wild type alleles, and then on the other chromosome 15 from mom, those are the expressed ones. And if those are mutated, you will get Prader-Willi. Right? However, if in the same region of the chromosome 15, if mom's chromosome region is imprinted and turned off, and dad's region is on but mutated, you then will have a child with Angelman syndrome. Right? Now, this is because these are different genes, right? They are very different disorders and they are due to different genes. The genes just so happen to be in a region that gets imprinted and turned off, right? This is the region that's getting silenced, right? And so if you've got the abnormal genes, then you're going to have the condition, right? So Prader-Willi, interesting situation, right? Okay. The last thing, linkage and recombination. Again, I've referenced linkage. I've talked about how if Mendel had studied two traits that were physically like right next to each other on the chromosome, he wouldn't have seen the pretty relationships that he did see with tall and dwarf, or yeah, tall and dwarf, or yellow and smooth, or yellow or green, smooth or wrinkled, right? Those either or, dominant recessive, 3-to-1 ratio, 9-to-3 to 3-to-1, that's all Mendelian. But when the genes are physically close to each other, Mendel goes out the window, right? So this is when linkage comes in, right? Now, it took a while to figure out what this was, and it started with observations of mating true breeders, right? True breeding parents generating heterozygotes in the F2 in the F1, just like Mendel, right? And then allowing the F1s to self-fertilize just like Mendel, right? But then when you looked at the F2, it wasn't the 9-to-3 to 3-to-1, because again, we're following two traits, right? We're following flower color and pollen shape, right? So when Mendel looked at seed color and seed shape, it was the 9-to-3 to 3-to-1 always, right? When Bateson did his studies with different flowers, different species, right? He came up with a wildly different ratio, 15-to-1 to 1.5 to 4.5, right? Not the expected. So something was going on, right? This is Bateson and Punnett, right? Punnett Square, right? They did this work. So that's interesting. What does that mean? Well, maybe these are, again, they're physically linked, but we don't know yet. We had to wait until Morgan came around studying fruit flies, and he figured this out, right? They only have 4 pairs of chromosomes, so it's pretty simple. Three autosomes and 1 sex chromosome pair, right? So it's a relatively simpler model system than a human cell or a cat or a dog or whatever, right? So what he did was he got his true breeders, the homozygous, dominant and recessive, so gray body, straight wings, black body, curved wings, and he mated them, all right? And all of the F1s were dominant, gray body, straight wings, were good so far. Then he did not F1 self-fertilization, but he did a test cross. I don't know what drove him to do that, but he did. So he did a test cross, right, to then produce the F2s. And this is what he found with the F2s. All right. He saw individuals that were, just give me a couple of minutes. Yeah. He saw individuals that displayed, so this is the F2, right? He saw individuals that displayed the same traits as the P generation parents, right? So gray body, straight wings, right? That was back here, or the parent, right? Parent, gray body, straight wings. Parent two, black body, curved wings, right? So he did his little test cross, and he found that there were the double dominant gray body, straight wings, right? This is what he expected. This is what he saw. Then there were a good number of black body curved wings. That's the double recessive, right? And then the really interesting thing was that he saw two new phenotypes. He saw flies that had the gray body and curved wings. And then he saw the other new combination of black body, straight wings. Right? And these ratios, these numbers were much smaller. Right? And so he termed the flies that looked just like the original parents, those were the non-recombinants. There was no recombination during meiosis of the genes in that cause body color and body shape. There was no recombination between those two genes in these two individuals because they have the exact same phenotype as the parents. However, in these two individuals, right, this is a new combination, right? So you can look at the chromosome B plus C, BC, right? There is no B plus C chromosome up here. So that's a brand new chromosome because of the crossing over. And then here's the other brand new chromosome, BC plus, right? So these crossing over events produced unique chromosomes, and that then led to unique new phenotypes. And he called those the recombinants. So something had recombined during meiosis, right? And so he looked, and these numbers were always not 9 to 3 to 3 to 1. Oftentimes it was like, what is it, 13 to 7, 9 to 7, I think is a common one. 9 to 7 is a common one, right? But the result of Morgan's work, right, with looking for offspring in the F2 that had the new combination of phenotype, right, by looking to see how frequently those new phenotypes would appear, what chance of the genes crossing over during meiosis so that we get those new recombinant chromosomes, those new recombinant phenotypes. The closer the genes are, the lower the probability of crossing over in between, right? The farther two genes are from each other, the higher the probability that a recombination event is going to occur, and then those two genes are going to be independent, even though they're on the same chromosome, right? And this probability was converted into what we call map units. And those are centimorgans after Dr. Morgan, right? And each map unit is a 1% chance of recombination, right? Which means, but a gene that's located at 0.0 and then a gene that's located at 13.0, there is a 13% chance that during meiosis, this chromosome is going to break here and then exchange this with its homologous partner, right? And then the homologous partner's chunk is going to come over, right? That's crossing over, right? So two genes that are kind of close, 13%, right? But that gene to that gene, more than half the time, right, you're going to get a crossing over event that separates this gene from that gene, right? Because they're going to move to the other chromosome. And then if you look at the bottom, right, 104, that's more than 100%, right? So that means that the chances of this gene ending up on a new chromosome compared to that gene is more than 100%. So almost always, all right? So the map units and linkage, it's based on the percent recombination chance. And the closer the genes are together, the lower the chance that we are going to split the chromosome between them during meiosis, during crossing over, right? So that's linkage, right? Right. So if we were to go back to use Morgan's data, right, the gray body and the straight, the body color gene and the wing gene, based on his numbers of recombinants, based on the total number, right, 27 map units apart, right? He saw recombinants about 27% of the time. So that was 27 map units between those two physical locations of those genes on one chromosome. So that's linkage. So again, we're not going to worry about that on the exam. I just wanted to get it out there for you. And same with imprinting, don't worry about it, but focus on X inactivation and then just epigenetics in general. Right? Yeah. So that is it for us. Again, I will send out emails with the testing information, but we will be in the Human Health Building, which is on campus. It's the northwest corner, the newer building, kind of by Squirrel and Walton, that corner. We will be there in room 4050, 4050, from 3:30 to 6:30 on Wednesday the 10th, right? So that's where we will be for our final. And again, I'm around. I'll be in contact, or I can be in contact over the weekend and whatnot. So if you've got questions and issues and whatnot, just send me an email, and I'll try to answer that. How much is the final worth? So the final is 120 questions, so 120 points. So it's really kind of worth two exams, if you will, but I don't weight it doubly because it's already twice as many points, right? But they just will get those 120 points are added into the total exam points, right? So I don't weight the final more, but it's double the points, right? 120, right? And then there will probably be some extra credit because why not, right? Yeah. Okay. Any other concerns or thoughts at the moment? I don't have a grade calculator available. I did update everybody's grade as of on Moodle for the letter grade. I have percentages with me in my grade book. If you would like to have an idea of what your percentage is overall before the final, send me a quick email and I will kind of send you what your percentage is overall, right? So I can do that for sure. Right? Yeah.
Is there any chance that we could like make a Zoom like appointment for next week so I could have like my exams looked over, my
Yes.
past first three exams?
Yes. So I have met with students and we have been able to look over exams virtually. So that can be done. I can pull up your Akindi sheet that's been scored, and then I can pull up the exam and we can kind of figure out which questions you got wrong, and then we can kind of go over those questions. So yes, absolutely, you can make an appointment and we can kind of review exams. Yep.
How do you make an appointment again?
So if you want to just either send me an email, right, that is asking for that, maybe give me some times, and then I can send you the link. Or if we go to Moodle, I'm going to go back to sharing real quick. All right. Hopefully, all right. God, I've got so much stuff going on. Let me get to where's Moodle? Okay, here's Moodle. All right. So this is Moodle. This is from my 920 class, but they're pretty much the same. If you scroll down to here, so virtual links for student success. Um, this one won't help you, but I will have my regular Zoom on Monday from 12 to 1. So that'll just be a general Zoom. I'll be open for that on Monday. But I can send you a link for a Zoom for an appointment at some other time. And then we can meet at that point and kind of discuss exams or whatnot. So if you want to make an appointment on Monday other than 12 to 1, and again, Monday and Tuesday mornings, I'm busy with exams. So Monday or Tuesday afternoons, if you want to set up a Zoom appointment, send me an email, give me a couple of times, and I will figure out what works and then send you a link. Yeah.
Okay.
Yep. Cool. All right. Anything else? No. Okay. So on Wednesday, I'll have the exam. I'll have the Akindi sheets. I'll have probably a slideshow, and I will have color hard copies of the images just in case there is no projector. I don't know what this room looks like, so we'll find out. All right. And all right. So have a great rest of your Friday and a good weekend, and good luck on all of your exams. All right. See ya. Take care.